Hello and welcome to our today's webinar. Today we will be discussing lipids in a very interesting context, namely context of skin research. Today we are hoping to provide an insight into lipid composition of the skin. We would like to highlight the role of lipids and particularly ceramides in the skin barrier function. And we would also like to touch upon the importance of skin lipidomics in studies that are related to development of diverse personal care products. So let's look at skin as an organ. And one has to remember that skin is the largest organ in the human body. It has a really complex structure and exerts a multitude of different functions. First and foremost, skin is our reliable barrier, which shields us from the outside world and prevents penetration of diverse pathogens and exogenous agents, xenobiotics, through the barrier into our body. On the other side, it also prevents excessive water loss and loss of other valuable nutrients and ions from our body. The skin is also considered an important immune organ due to its critical role in maintaining barrier against microbes and other potential intruders. It also contains a really finely tuned interaction between immune and non-immune cells that are mediating processes like wound healing, inflammation, and allergy. Skin is also a very large sensation organ. It contains Merkel cells, which are functioning as receptors and are responsible for stimulating sensory nerves that brain perceives as touch. These cells are especially abundant on the surfaces of hands and feet in the human body. Last but not least, skin is an important biosynthetic organ. Substances like vitamin D and melanin are synthesized in the skin as the response to UV irradiation. So skin anatomy is important to understand when speaking about skin lipids. Barrier function of the skin resides mainly in stratum corneum, the outermost layer of skin, and particularly composition and organization of the lipid matrix in stratum corneum, which mainly contains ceramides, free fatty acids, and cholesterol, are of importance. We would like to start our webinar with a deeper look into skin morphology, and we would like to touch upon the origin of lipids in epidermis. Histologically, skin consists of epidermis and dermis. And today we will be mainly speaking about epidermis, which starts from basement membrane that separates dermis from epidermis, and the first layer of cells is called stratum basale. Before we look into each individual layer of epidermis and its importance for skin lipid composition, let's take a look at one additional structure in epidermis, which is called sebaceous or oil gland. This is also a contributing factor to skin lipid composition, and we will be talking about this a lot today. So sebaceous or oil gland is a structure which is consisting of cells which are called sebocytes, and they are specialized in producing and secreting sebum or skin oil. This secretion is then drained into hair follicle and is secreted towards the surface of the skin, where it exerts additional protection and hydration function. And this oil secretion consists of free fatty acids, wax esters, triacylglycerols, deacylglycerols. So now let's take a closer look at the life cycle of keratinocytes and the process of their differentiation, because this is particularly important for skin lipids. So differentiation or life cycle of keratinocytes begins in the basal layer, where These cells are differentiated out of stem cells. The stratum basale exclusively consists of basal cells. This is a single layer of cuboidal shaped cells, which are constantly going through mitosis to produce new cells. As these cells are formed, existing cells are pushed superficially away from stratum basale. The next layer is called stratum spinosum, or spiny layer, and it has a very specific histological appearance due to protruding cell processes, which are called desmosomes. And these are the structures that interlock with each other in order to strengthen the bond between the cells and create an additional layer. Spiny layer consists of 8 to 10 layers of keratinocytes, and the cells in stratum spinosum begin the synthesis of keratin and release glycolipids, which are essential for skin barrier function. As new keratinocytes are produced atop of stratum basale, keratinocytes of stratum spinosum are pushed into the next layer, which is called granular layer or stratum granulosum. This layer is called this way because it has a grainy appearance due to the further changes that occur in keratinocyte. And you can see here in this slide, these cells are smaller, flatter, and they contain specific granules inside. The membrane of these cells thicken, and large amounts of keratin and keratohyalin are produced. And these substances accumulate in lamellar granules, which we will discuss a bit later in this webinar. Nuclei and other cell organelles disintegrate in these cells, and cells die, leaving behind keratin, keratohyalin, and cell membranes that will later form stratum lucidum or stratum corneum, depending on localization on the human body. Here you can see the next layer is called stratum lucidum, but I have to mention that this layer is not universally present in all parts of the body. This is a seemingly translucent and smooth layer of epidermis, which is located just above the granular layer and below stratum corneum. This thin layer of cells is only found in so-called thick skin, acral skin, on the digits of the fingers, on the palms of the hands, on the soles of the feet, but also in different pathological conditions which are accompanied by excessive rubbing or scratching. There is a possibility, if this is happening for a prolonged amount of time, that stratum lucidum would also form in that areas. Keratinocytes that compose stratum lucidum are dead and flattened, and these cells have their appearance because they are densely packed with lipid-rich matrix, which is called alleidine, which gives these cells their specific appearance and also provides an additional barrier to water. And last, but definitely not the least in this chain, is the outermost layer of epidermis, which is called stratum corneum, which contains the most differentiated keratinocytes, or they're also called corneocytes because these are completely keratinized or cornified cells, which are dead. It consists of 15 to 30 layers of cells. This is a dry layer which prevents penetration of pathogens and prevents underlying tissues from dehydration. It also provides a very good mechanical protection to more fragile and delicate underlying layers of epidermis. Cells of these layers are shed periodically, and full renewal of stratum corneum takes up to 28 days or 4 weeks. Now let's speak a little bit more about stratum corneum and its lipid matrix. Stratum corneum is a complex matrix which is primarily built from protein. It's 75 to 80% of protein and contains around 15% of lipids. The architecture of stratum corneum can be represented as a brick and mortar system where dense enucleate protein-rich flat keratinocytes are presented as bricks, and intercellular lipid matrix, which has a very peculiar structure, is a mortar. In contrast to phospholipids, stratum corneum contains a lot of ceramides, which have small polar head and a very long saturated chain with prevailing all-trans conformation. This hairpin structures that you can see on this slide representing lamellar organization of this mortar between corneocytes has a lateral packing which is called orthorhombic packing, and it's very dense. It has a highly limited motional freedom. There are also other types of lipid packing present in stratum corneum, namely hexagonal, which is less dense, and liquid, which is not bordered. And the way the lipids are arranged in stratum corneum is directly influencing the integrity of lipid barrier and transepidermal water loss that can occur. Okay, now let's speak about the interface between stratum granulosum and lipids. So as we have seen before, a granular layer contains lamellar granules, or also they are called Odland bodies. And these are organelles that appear in upper spinose layer, but are very peculiar for granular cell layer. All nucleated layers of epidermis are active sites of lipid synthesis. And ceramides are built in all nucleated epidermal cells. This is not the same for cholesterol, which is another prevalent component of stratum corneum lipid matrix, because cholesterol is synthesized in the lower cell layers, mainly in stratum basale. Synthesis of sphingolipids increases with epidermal differentiation, and therefore the most undifferentiated layers of epidermis mostly contain membrane lipids such as phospholipids, while lamellar bodies of differentiated keratinocytes are mainly filled with glucosyl ceramides, cholesterol, and phospholipids. Ceramides are synthesized de novo with involvement of an enzyme which is called serine palmitoyltransferase, which catalyzes conversion of both palmitoyl-CoA and L-serine into ceramides in epidermis. And during their synthesis, the different ceramide species are modified at their 1-hydroxy position to either glucosyl ceramide or to sphingomyelin. Following sequential formation and sequestration of ceramide precursors, they are secreted then from lamellar bodies into an interface between stratum granulosum and stratum corneum, where they undergo extracellular hydrolysis to free ceramide species. Ceramide species are generally formed via 2 pathways. The first pathway is through degradation of glucosyl ceramides, which is catalyzed by beta-glucocerebrosidase, and the other pathway is including hydrolysis of sphingomyelin, which is catalyzed by sphingomyelinase. Okay, to summarize the first part of our talk, we have spoken about different structures in epidermis that play a role in production of skin lipids. We have spoken about sebaceous glands and sebum, and shortly about its composition. You know that sebum is essential for skin protection and skin hydration, and is also implicated in oily skin and acne. We have spoken about progression in keratinocyte differentiation and how this is connected with lipid synthesis, modification, and excretion. And I remind you that stratum corneum is the most important part of skin barrier, which is responsible for skin hydration. This is the layer which is impaired in a multitude of different diseases and skin conditions like dry skin, atopic dermatitis, sensitive skin. And this layer is also important for cosmetic product penetration. In this section of our webinar, we will speak about Lipotype Skin Lipidomics platform, and I will present data from several studies which have been supported by the data generated using our platform. The skin lipid profile is affected by a multitude of different factors, such as age, sex. There is a certain seasonal variation with depletion of skin lipids during the winter time or during the cold season. There is a variation of skin lipidome depending on the depth of skin sampling, and we will touch upon this topic in the next study. And there is also a variation depending on sampling site. There is also such thing as skin type, which is partially dependent on our genetic makeup. And of course, skin diseases also alter skin lipid composition. In turn, one could also say that skin lipid profile influences multiple functional parameters of the skin, such as skin hydration, transepidermal water loss, surface pH, microbial composition of the skin, and also skin morphology. On this slide, you can see sampling of stratum corneum lipids using D-squame tapes. And this is the typical procedure that we are using in order to sample skin lipids from the patients. These tapes are small, flexible plastic discs, which have an adhesive surface that is placed onto the skin, and it allows to remove a layer of stratum corneum. This enables painless, non-invasive, and reproducible sampling of up to 20 layers of stratum corneum. So let's imagine this small D-squame disc with stratum corneum has been sampled. What happens next? In the first step, we will extract this tape, and we will add internal standards to the samples for the purpose of lipid quantification at a later stage. Afterwards, these lipid extracts are directly infused into a mass machine, and a mass analysis takes place, which leaves us with a very large data set, which consists of all possible lipid data that has to be annotated, identified, and quantified. And for that, we do have two proprietary software platforms, one of which is called Lipotype Explorer, and we perform lipid identification using this platform. And the other platform is called Lipotype Zoom, which allows quite a sophisticated data reporting. Our method is designed for untargeted broad coverage analysis with absolute quantification, as opposed to other methods that offer relative quantification. This is a method that has high technical precision and allows high throughput. We have the capacity to analyze thousands of samples a week. This is also a method which has a possibility to identify lipids very fast. It enables quite a sophisticated structural reporting, so some lipids, depending on the class, can be reported on subspecies level. And this method is particularly suitable for large-scale clinical lipidomic screenings and cohort studies. I would like to present a study which was conducted by Tomasz Sadowski and the team of Lipotype back in 2017. And this study was primarily focused on validation of a skin lipidomics platform, and it also dives deep into inter-individual and intra-individual variability of skin lipidome. So around 300 samples were harvested in this study from 104 participants. And this slide represents a coverage which could be yielded in the process of method validation. So we can cover more than 400 individual lipids, which belong to 16 distinct lipid classes. These are 12 subclasses of ceramides. And we will touch upon the classification of ceramides in a later part of the presentation. There are diacylglycerols, triacylglycerols, which are classical sebum lipids, as well as cholesterol and cholesterol esters, which can be identified. The next slide represents the linear dynamic range of the method. So there is a linearity across 4 orders of magnitude, which makes this method particularly suitable to measure samples where we expect both very highly concentrated lipids and also lipids with very low abundance. In this study, the impact of age and sex on variability of skin lipidome was assessed, and for that, 65 female and 39 male participants in the age range between 20 and 89 were included into the study. So the total lipid amount in males did not correlate with age, but in females, there was a negative correlation between age and total lipid amount. When we look more precisely into compositional changing, which lipid classes contributed to this change, we could see that relative amounts of sebum lipids, triacylglycerols and diacylglycerols in female skin markedly decreased with age, while the relative amount of cholesterol increased. These changes were less pronounced in the male study participants, and a possible explanation for gender-specific age-dependent depletion of sebaceous lipids might be connected to hormonal regulation of sebum production. It was shown in previous studies that increasing age is either reducing the density of eccrine glands or their functionality. This could account for the pronounced decline of both absolute and relative amounts of sebaceous lipids among older women. However, follow-up study with a larger number of participants and more strict confirmation of their hormonal status might be necessary. Now let's move on to differences in skin lipid composition depending on the sampling site. To assess intra-individual lipidome variability, the sampling was performed in triplicates from 14 different body sites, as shown on the slide, from 1 female and 1 male participant. Across all sampling sites, we observed an intra-individual variability of total lipid amount of 104% for male participants and 81% for female participants. And the most distinct samples for both subjects were from forehead, where we have observed total lipid amounts as high as 21,000 picomoles per sample, and from the heel, where this amount was as low as 300 picomole on average. Such differences in the lipid content of skin from various body parts are not sensational. This has been published before. You know that different zones of the body are traditionally classified to dry and moist zones. They have different microbial composition. They have different density of sebaceous glands on them. They have different exposure to outside environment. Therefore, these compositional changes can be very well explained. Now let's take a look on the influence of sampling depth on lipidome composition. And to check how skin lipidome changes with the depth, sequential tape stripping until 20th layer was performed on the left volar forearm in 1 female and 1 male participant. In both participants, total lipid amount continually decreased by 85 and 87% until the 5th and 7th layer of sampling for female and male respectively, reaching a plateau thereafter. This could be explained either by higher mass of skin sampled from the topmost layers, or by a higher proportion of lipids when compared to other constituents of stratum corneum within these layers, or actually by a combination of both. Observed surplus in lipid amount in the uppermost layers compared to deeper layers is greater than would have resulted from reported differences in sample weight. And this suggests that the uppermost layers, while more abundant in mass, are enriched in lipids compared to deeper samples. Topical excretion of sebaceous lipids could also account for this observation, as sebum lipids were shown to surpass stratum corneum lipids on the skin surface over 10-fold by weight. Again, if we look at compositional changes, if we look at particular lipid classes and how do their amount change with deep sampling, in both subjects, we have seen marked decline in relative amounts of sebum lipids, triacyls, glycerols, and diacylglycerols, here represented as an orange line. And jointly, these lipids account for half of known sebum lipids on average. Therefore, this decrease, when combined with decrease in total lipid amount, makes sense. Relative amounts of cholesterol and all ceramide subclasses increased with sampling depth. On contrary, ceramides, as we discussed before, are synthesized deeper within stratum corneum and are not a constituent of the sebum, which fully accounts for observed ceramide sampling depth gradient. Likewise, cholesterol constitutes as much as 1/3 of stratum corneum lipids and only a small fraction of sebum lipids, which is also reflected in our data and its lower relative amount at the skin surface. Similarly, cholesterol ester was previously reported in stratum corneum as well as in sebum, and this is confirmed by our measurements where relative amount of cholesterol esters decrease in the topmost layers and increases again below 5th, 7th stripping layered. In summary, we have observed age and sex dependent variability of skin lipidome. Skin lipidome is highly body site specific, and there are also peculiarities related to sampling depth. Now, in the next study, I would like to present connection between skin ceramidome and skin pigmentation. And to start this part of the presentation, I would like to mention that the data on ethnic differences, especially in facial skin, in relation to biochemistry of stratum corneum are very scarce, fragmentary, and often contradicting due to the fact that a lot of studies are still performed in Caucasians. They include a very small number of participants, or they utilize a diverse methodology which cannot be compared to each other. However, we have tried to summarize published data on skin differences in different ethnic skin. I would like to emphasize that people with darker skin tone as consumer group, they tend to have dry skin concerns or ashy skin or skin desquamation as a concern more often in comparison to their Caucasian counterparts. And this is a legitimate skincare concern, which is addressed by cosmetics market. There is literature which suggests that black skin type has more cell layers in stratum corneum compared to white skin. It also has higher rates of spontaneous desquamation compared to white skin type and East Asian skin type. Black skin type shows also slower desquamation rate when compared to white and East Asian skin types, and black skin type has higher dryness score when compared to white skin type. In the next slide, we would like to touch on the ceramide classification before we move on to the next fragment of the study that we would like to present. Here on the slide, you see chemical structures of 12 ceramide subclasses, and each ceramide molecule is composed on sphingosine base and a fatty acid. So this classification is based on molecular structure, which corresponds to sphingosine base. You see horizontally, these bases are dihydrosphingosine, sphingosine, phytosphingosine, and 6-hydroxysphingosine. And there's also a fatty acid component. So this way, a molecule which is composed from 6-hydroxysphingosine with alpha-hydroxy fatty acid is denoted as ceramide AH. Now let's move on to the study. This is a study which was published in 2022, and it was supported by the data generated with our platform. The study was a cross-sectional study which was conducted in South Africa, Pretoria. It included 60 healthy female volunteers, which were divided into 3 age-matched groups, 20 women per group. So these women were of albino African origin, Black African origin, or Caucasian. Participants were required to not apply any cosmetic products onto their skin 3 days prior to the study. And out of the whole study pool, 24 participants were selected for lipidomics analysis. Here you can see the sampling site where tape stripping has been performed. These were tape strips taken from the cheek and also in post-auricular area behind the ear. Lipidomics analysis revealed that total ceramide levels in different groups were quite different and quite surprising for one of the subgroups. There were no significant differences in total ceramide levels or ceramide subtypes between Black African and Caucasian women, either on the cheek, neither on the cheek or nor on post-auricular samples. However, compared to other two groups, the levels of total ceramides as well as the levels of omega-O acyl-ceramide species were increased in albino Africans, and this was a significant increase. On this slide, you can see the subclasses profile for ceramide composition, and there was a significant elevation of ceramides AH, AS, and EOH when compared to facial skin from Black Africans and Caucasians. And these findings were quite unexpected, as I said, because these participants very often suffer with impaired skin barrier function. However, elevated levels of omega-O ceramides can contribute to barrier insufficiency per se. You remember the slide about lateral packing and lamellar organization of the lipids. So imbalance in certain ceramide subclasses can impact stratum corneum lipid phase behavior and can reduce actually the barrier function. Additionally, these differences in the levels of omega-O acylceramides were connected to another study where the researchers measured the amounts of acylceramide processing enzymes, namely levels of arachidonate 12 lipoxygenase and short-chain dehydrogenase or reductase family 9C member, which were increased in albino Africans in comparison to Black African and Caucasian women. Induction of these enzymes is most likely a compensatory mechanism to correct for impaired process of lipid envelope maturation in corneocytes in this subgroup of people. Small summary of this study. Lipids, particularly ceramides and stratum corneum, are essential component of skin barrier. And non-pigmented albino skin with impaired barrier function reveals higher total ceramide levels and increase in acyl ceramides. This increase is related to changes in the epidermal ceramide processing enzymes. Moving on to the next study, we will briefly touch upon the topic of ceramide composition and lipid composition in skin, which is affected by different diseases. So we have already discussed classification of ceramides. Now let's take a look at this slide, which is a brief overview of skin lipid composition in different conditions and diseases. I think it's also worth to prepare a next webinar, which is entirely focused on skin lipid composition in atopic dermatitis or psoriasis. So we will only be briefly touching this topic in today's webinar. In atopic dermatitis, skin lipid composition is altered in terms of decreased total amount of lipids, and particularly ceramides are affected. The most affected ceramide subclasses in this disease are acyl ceramides, ceramide EOS, which is profoundly decreased in atopic dermatitis. Studies on atopic dermatitis also revealed that not only the subclass of ceramide is altered in atopic skin or lesional skin, but also the chain length of ceramides is reduced. So ceramides with chain length of 34 are considered to be short chain ceramides, and they are increased in lesional skin, whilst the long chain ceramides, which are actually superior in their hydrophobicity, in their possibility to build this densely packed mortar between corneocyte, they decrease in the skin of these patients. And these alterations in lipid composition are also correlated with high transepidermal water loss and the symptoms that patients experience. So let's move to the next study, which is actually a nice illustration of how claim support for different personal care products can be performed in vitro models using lipidomics. And in this study, glycerol petrolatum-based emollient was applied on reconstituted human epidermis model. And this application was repeated on days 9, 10, 13, and 14, and on the day 15, the samples were harvested and snap frozen for downstream lipidomics analysis. And you can see on this slide, a significant impact of the cream on total ceramide content was observed in treated epidermis when compared to control, and this was an increase of 38%. Not only total amount of ceramide increased after topical application of the cream, there was a significant increase in ceramides EOS and EOP, and it's worth mentioning that acyl ceramides play a very important role in the structural integrity of the skin, with ceramide EOS being an essential component creating and maintaining lamellar lipid structure. A reduction in the level of EOS results in a change in the orthorhombic structure of epidermal barrier to a more hexagonal and liquid structure, which increases skin permeability and enhances transepidermal water loss. Furthermore, we would like also to show the result which demonstrates that there was a significant increase in short chain ceramides, ceramides which are defined as ceramides with carbon chain lengths of C34 till C44, and there was a statistically significant decrease in treated epidermis. At the same time, there was an opposite trend observed for long carbon chain length ceramides, which we're not showing in this presentation for the sake of time, with significantly higher quantities of these ceramides in cream-treated epidermis. Very short summary about emollient application and changes in ceramidome. Skin diseases are associated with reduction in total amounts of ceramides in the skin and also reduction in long-chain ceramide species. In vitro application of emollient leads to an increase in total ceramides and particularly a significant increase in EOS and EOP subclasses. And this treatment also has proven to have a beneficial influence on ceramide chain length. Final study that I would like to present today is related to dry skin, and dry skin is a concern for approximately 30% of healthy population. In this study, participants with cosmetically dry skin were allocated to 5 weeks of application of moisturizing lotion, which contained free fatty acids and small amount of occluding agent. And there was an investigation on the baseline, which contained clinical investigation. There was lipidomics, samples taken, and there was also microbiome samples taken from participants, but we will not be looking into microbiome aspect because this is definitely another topic for a separate webinar. The same investigation has been performed after 5 weeks of moisturizer application, and as a result, one can see in the changes in total level of free fatty acids pre and post intervention. These changes were measured in outermost 5 tape strips. On the slide, on the graph B, you can see changes of cholesterol level prior and post intervention, which were measured in outermost 5 tape strips and also in the deeper tape strips from layer 16 to layer 20. And one can see that while cholesterol levels in strips, in the upper strips did not change, in the deeper strips, there was a significant increase in cholesterol after 5 weeks of lotion application. The next slide illustrates ceramidome changes prior and post intervention. So there was a significant increase in ceramide classes ADS, AH, AP, AS, EOH, NH, NP, and NS post intervention. There was also a very nice correlation of ceramide values with corneometry values, which are representing skin moisturization levels. We're not showing these results here for the sake of saving time. So, small summary. Applying body lotion to cosmetically dry skin can improve skin hydration, and the effect of this intervention can be quantified. Changes in ceramide composition, free fatty acid composition, and cholesterol are detected upon treatment with body lotion. I would like to summarize the whole presentation. Today we have learned that skin lipids are major contributors to skin barrier function. Skin lipidomics can elucidate the relationships between skin lipids and skin function. Skin lipid profiles vary within and between humans and also depend on sampling depth and sampling site. Skin lipid profile and ceramide composition is altered in skin diseases. Skin ceramide composition is changing upon application of moisturizing lotion, and the result can be quantified.